I've added to your shared Google Sheets a number of new pages. They're titled SNP Info, Day Zero SNPs, Day Zero Indels, and then your group's final sequencing data, SNPs and indels. So I'm going to walk you through all of these pages and kind of what we're looking at. All right, so the SNP info page is where you're going to keep track of all of the mutations that you're analyzing in depth. So SNP stands for single nucleotide polymorphism, and indel stands for insertion deletion. And those are the two main groups of mutations. And so this is going to be your list of mutations that you're looking at to see what they do to the genes or promoter regions and what those might mean. So I'm actually going to come back to this page after we kind of look at the original data. So let's hop over to the Day Zero SNP pages. All right, so the main columns that we care about are column B. POS stands for position. So this is the location in the genome of your mutation. So when you are looking at the E. coli genome that you imported into your benchling, this is its identification number. It should look a smidge familiar. You copy and pasted it once to import this into benchling. This is literally the base number. So you would navigate in benchling to base number 1043745. And yes, that's a lot of scrolling. Ref stands for reference. So this is the base that is present in the reference genome. So that's the base that you're going to see on benchling in that location. Alt stands for alternative. So this is the alternative base that was present in the sequencing data. So effectively, this is the mutation. So it started as an A, and it was mutated to a T at, again, base number 1043745. The only other thing we're going to look at from this page is way over here. So this column I says format, and it's telling you the formatting of the numbers in column J that are specific to your sequencing data. And again, the only one we care about here is AD, which is the second one after the first colon. AD stands for allele depth. This is what is going to tell you what percentage of your reads, your sequencing reads, had this mutation. So if we look over here, I'll try to highlight so we can see what we're looking at. We're looking after the first column, because that first one is not what we want. We want after the first colon, sorry, AD. So those are those two numbers right here separated by a comma. These are the number of reads that have your reference and then your alternate. So in this particular mutation, which I'm just using as an example, 0 reads had the reference A, and 355 reads had the alternate T. So remember what the sequencer actually did was take all of the many copies of the chromosome from all the different cells in your population and break it up into little pieces and then sequence each one, right? So we have a lot of reads because there were more than one cell and more than one copy of the chromosome in the sample that we sent. And so this particular base got sequenced 355 times. 0 times it had the reference A, and 355 times it had the alternate T. So those numbers, the 0 and the 355, in addition to position, reference, and alt, are what you may bring over to your SNP info page, right? Here's the position, the reference base, the alternate base, and then those two AD numbers. So again, the first number, which is the number of reads with the reference base, and the second number, the number of reads with the alternate base. And what you can infer from these two numbers is what percentage of your reads had your mutation, right? So the total number of reads are these two numbers combined, and the number that have been mutated are the second number, And so you can easily figure out a percentage from that. And that's your proxy for what percentage of your population was carrying that mutation. So again, in the one I looked at as an example, 100% of the population seems to be carrying this mutation because we didn't see any reads with the reference. But that won't always be the case. You'll often see a mixture where it's not 100%. Again, indel stands for insertion deletion. So this is where we're taking one base and changing it to a different number of bases. As an example, just looking here at the first row, our reference is T and our alternate is TG. So that means we've inserted a G effectively by adding an extra base. And now if we come over here and find our allele depth, we see we don't have a 0, right? We've got 6 and 245. So 6 reads that had the reference, just T, and 245 reads that had the alternate, TG. So this is in the majority of the population, but not quite 100%. All right, so now why do we have 4 pages of mutations and not just 2? So these first 2 pages are day 0, and these last 2 pages are the final population, so roughly day 21, specific to your group. I have different little notes at the top of each of these pages to help you understand what to do. Let's start with the easier one to understand. So this is group 1's SNPs. So these are the SNPs in their final population. So if any of these are not in the day 0 list, highlight it and copy it to the SNP info page. Okay, why would that be the one we're looking for? All right, so day 0, I'm going to hop back over here for just a second. These 4 SNPs are differences at day 0 from the reference. So remember, the reference genome is just the database genome, right? These are a standard E. coli sequence, and it was supposed to be really close to the E. coli that we used, and indeed it was, right? We didn't have a long list of differences between our specific E. coli that we used and that reference genome in the database. But again, these are our differences from the reference at day 0. So effectively, these are what looks like a mutation to the sequencer and to the algorithm that we use to analyze our data, but they're not really mutations we care about. These were already here from the very beginning. So that's why it says on your SNP page, if they're not on the day 0 list, then we care about it, right? Because if it was already there at day 0, it's not a mutation that we actually care about. It didn't happen during your experiment, right? It was there when we started. But the ones that are not on the day 0 list, that would mean the strain was originally the same as the reference on day 0, but in your population, a mutation changed it to the alternate sequence indicated, right? So we want to take those mutations and move them to our SNP info page for further analysis. We're going to do the exact same thing with the indels. This list is a little bit longer. We've got 41 in this particular data set compared to 34 that we already had at day 0. So again, most likely what happened is there are the same 34 at the end that we started with, but there's an extra handful in here. So you need to cross-reference between the 2 lists, find the indels that are new in your specific evolved population that were not there at day 0, and then only analyze those. The other thing you're going to do is look through the day 0 lists and see if any of those are missing from your final population. So I have these notes at the top. If any of these are missing from your list of SNPs, highlight it and copy it to the SNP info page, right? We care about that because that would mean that the strain was originally different from the reference, but in your population, a mutation changed it to now match the reference. And because it now matches the reference, it doesn't show up as a list of mutations. And so if any of these pre-existing differences from the reference get lost, again, those are changes that happen during your experiment that we care about. So you need to cross-reference between the lists in both directions. look for any of the day zero SNPs or day zero indels to be lost. We care about those. And obviously we care about new SNPs and new indels that arose during your experiment that were not there at day zero. One thing that's a little bit tricky is if you have one that gets lost, you need to reverse the reference and the alt columns. Because effectively, you started with the alternate base at day zero, and you went back to the reference over time. And so you'll want to flip-flop those for the purpose of looking at what changed in the protein. All right, that's it for this, and I will come back with something else later potentially.